Kate, can you let us share our video instead? She already did. Okay, good. I just need to make this thing go away. Okay, it should work fine. Okay.
All right, welcome to everyone in the room and to the audience that I assume is listening in on Zoom as well. It's my great pleasure today to introduce our speaker from the Division of Nephrology, Dr. Alexander Bullen. Dr. Bullen did his medical training at the University of Panama and then his internal medicine training at the University of Texas in San Antonio. We were incredibly fortunate to recruit Dr. Bullen here for his research fellowship starting in 2015 when I met him as part of his 3-year fellowship in nephrology. He came to us with a huge amount of energy and creativity, and I vividly recall the first project that we worked on together, which was actually a video game to teach people about hypertension management. He went on to do some really nice work in our dialysis unit, looking at individualized cool dialysate projects. And it was no surprise to anyone that he was offered both a career development grant from the NIH and another one from the VA. And having only to choose, he chose to work here with us at the VA. So it's actually quite appropriate that today's medical grand rounds is here at the VA. Where Dr. Bullen now holds a career development award and studies the effect of biomarkers of tubular function in kidney disease. So I'm really pleased to welcome Alex Bullen here to give us medical grand rounds.
Thank you to everyone for being here. Hopefully everybody got their code 8620, very important. So we'll talk today about APOL1-mediated kidney disease, a long journey toward precision medicine. I do not have any disclosures. So I was supposed to have this slide to talk a little bit about myself. Dr. Rifkin already did that for me, so this will be very quick. So the first picture here of the Panama Canal, like Dr. Rifkin said, didn't I mention I was born and raised in Panama. Grandparents came from the Caribbean, went there to work in the Panama Canal. Then Texas, San Antonio for internal medicine, and then San Diego, taken with my iPhone here. So that's in the trails in Rancho Bernardo, just running. So we'll talk a little bit about the background, then we'll go into the pathophysiology and then treatment about this very interesting disease that's kind of been known since 2010, kind of reaching its 15-year anniversary. So one of the things that I also do, I'm the course director of a class, an elective course for the medical students. It's called Fade Hypertension, so the fade of the haircut, the idea of the fade. And essentially, I talk to the students about blood pressure management, and we go to the community. We go to barbershops. This is one of them. So we go to barbershops. We also kind of go to places, mosque. There's the Malcolm X library. We go to the community essentially, and we talk to the customers and the people that we see about the importance of blood pressure control. And there's a small study that we're doing. Also give the patrons there, customers, we give them blood pressure machines. And in this particular study here, this is one of the barbers kind of teaching them about this and talking to them about that. As part of that role as the course director of that class, I was invited to be a panelist on an event by the Jedi community. Essentially, we went to the community and we discussed about different activities that UCSD can do to kind of connect more with the community. There speaking is the Vice Chancellor, Dr. Carruthers, the CEO, Patty Mason was also there. But the reason why I put this here is that there was a slide that really got my attention, where essentially they mentioned how our two centers, UCSD Hillcrest and La Jolla, the life expectancy, someone is born at Hillcrest versus being born in La Jolla, the difference that in life expectancy was 10 years. I was not expecting to see that. I could have guessed, as someone that works in both sides, and many of you want to be really honest, we will not be completely surprised about the life expectancy, but not 10 years. That seemed quite remarkable to me, like the same group of physicians, access to the same facilities, all the technologies and all that, and seeing that big, huge gap between the two centers. Then you get into a little bit into trying to understand this, you kind of think about the southeast of San Diego, the different limitations that there are there. There was an area known as the Four Corners of Death between Imperial Avenue and Euclid Avenue and in the Lincoln Park. So there are some reasons to understand that, and some of kind of this picture here is saying that south of the 8 is not just a location. There's a lot of problems that happen in that area. Some are kind of local, some exposures from the government, other things. This picture here, Varios C, Yonkers Now, essentially how the freeways that were made, the Varios is misspelled with the V, so it's supposed to be the B, but we'll leave that aside. So basically they're saying neighborhoods, yes, young yards, no, because that's what was happening when all those freeways were put there and kind of dividing the neighborhoods there. So if we look at that, there are many clinical, social demographic factors that kind of contribute to that disparity, health disparity, high school education, and the jobs, the food deserts, all the things, environmental pollution, chronic disease rates. And when we get also, so the point here being that whenever we look at differences between the whites and the blacks or African Americans, there are many reasons that have been posed and many that we can understand that exist. The same applies with kidney disease. So even though African Americans constitute less than 15% of the population, almost a third of the people on dialysis are blacks. And one of the main reasons that has been given to that, besides the other factors that I mentioned before, has been because of hypertensive nephrosclerosis. So essentially hypertension driving kidney disease. Since 1977, I'm showing here in this timeline, there was a study done in different counties in Michigan. And what the investigators were able to find is that there was a higher prevalence of hypertension. So they kind of mentioned this possibility of high blood pressure leading to kidney disease, kind of seeing this disparity. So 160 per million of blacks compared to around 40 per million whites. So a fourfold kind of risk among blacks compared to whites. But already by 1988, this kind of was being disputed that the racial differences were not fully explained by hypertension associated kidney failure, that there must be something beyond that. In 1993, Barry Friedman and his group published in the American Journal of Kidney Diseases, they looked at several generations of people that were on dialysis, and they kind of posed like there must be a component possibly, or there's the possibility of a genetic factor. There must be something more than just the idea of hypertension. So we fast forward to 2002, and then it's published in the Journal of the American Society of Nephrology, kind of the top journal in our field. And kind of they did a very elegant study looking at the excess risk that could not be explained by all these socio-demographic factors. And that same year, there was another study published by Kidney International, and I highlighted this part here saying that blood pressure levels and proteinuria did not account for morphological lesions, suggesting other factors, and they mentioned there such as genetic factors, microvascular disease may play a role. So there was some inkling about that already in the early 2000s about that role. So in 2008, there's a gene that's identified MYH9 that was associated, they posed that with non-diabetic kidney disease among African Americans. So there are two genes in the chromosome 22, the MYH9, and also in 2010, we learned about the APOL1. But then by studies done by Genovese and led by the group of Martin Pollack in Harvard, they're able to kind of isolate doing regression analysis and comparing to using the Genome Project and looking at databases from Africa. They were able to kind of isolate it more to the APOL1. So what is the APOL1? It's a component of the innate human system. It's found in HDL particles. And it's a component of, there are 7 forms of the APOL. The APOL1 is different from the other ones in that it has a signal peptide that allows it to kind of leave the cell being found and then in the serum. One of the important things to mention about the APOL1 is that it's not found in non-primates. And among the primates, our closest closest primate, the chimpanzee, already has it lost. So it's believed that appeared approximately 10,000 years ago. So looking at a little bit at the evolution of the APOL1, this, as I'll mention more later on, is a trypanolytic factor. So the presence of the APOL1 kind of prevents the infection or destroys the parasite known as Trypanosoma brucei. Then the Trypanosoma brucei kind of evolved and now we have 2 subspecies, the Trypanosoma brucei gambiense and rhodesiense. So the rhodesiense is found more in East Africa, the gambiense found more in West Africa. Obviously, there could be a combination. So there in the middle, you can see Uganda, so both can be present. So the Trypanosoma brucei evolving to this subspecies, the rhodesiense and the gambiense, then is able to continue with this trypanolytic or avoid that the trypanolytic factor of the APOL1, kind of showing here how it kind of it creates spores created and then leads to lysosomal swelling and lysis. So again, this is led by the APOL1. But by this mutation that the Trypanosoma brucei has evolved into the rhodesiense and gambiense, is able to avoid that kind of lysis, all that process that occurs with the presence of the wild type. Obviously, by now, this APOL1 mutation is beyond Africa. So other places that have African ancestry, so in Brazil, for example, there is a presence of that. In Panama, Jamaica, Grenada, so many other places. Obviously, the US has already been reported the presence of the APOL1, so kind of the evolution there. Now, going into APOL1 and the kidney. So first in 2011, so in 2010, when this is all identified, then in 2011, we kind of learned where exactly in the kidney do we find is the APOL1. So we know it affects the podocytes, the arterial endothelium, and also there is some evidence also of the proximal tubules as well. Important to mention, we don't know about exactly what APOL1 does in the kidney. Its presence is not necessary. In 2012, there was an interesting case of a man in India that was found to have Trypanosoma embansi, another trypanosoma. And when they did, so he had this infection. So different investigators kind of went there because we don't typically see this infection. He didn't have HIV or other risk factors. And when they did all these genetic studies, they found that he did not have the APOL1. So he was null for the APOL1. And they looked at different family members. And when they did all kind of looking at the usual metrics that we use to study the kidney function, looking at the BUN, serum creatinine, all those things were essentially normal. So again, we don't fully understand. And there have been other cases reported as well. So the presence of the APOL1 is not necessary essentially for the adequate function of the kidney. We know now that there are two variants of the G1 and the G2 that have appeared in the area where the Trypanosoma brucei had that modification in the serum resistance associated protein binding domain in the C terminus. So this is, so I don't know if you can see the cursor. So this is where the mutation happened that led to the Trypanosoma brucei, rhodesiense, and gambiense to be able to avoid or escape the trypanolytic factor of the APOL1. So by doing this mutation there, then by evolution, like I said, being reported, two risk variants appeared, the G1 and the G2, that allow the trypanosoma to still be destroyed, still create those pores, and then essentially be killed. So here in the bottom here, the Trypanosoma brucei rhodesiense. So when their presence of the G2, so two G2, G1 and G2, they are protected against the human African trypanosomiasis. Not so much for the gambiense, although it tends to be kind of more of an asymptomatic, but they can still be susceptible to that. as I'll discuss more in a few minutes, those same risk variants, the G1 and the G2, do increase the risk of chronic kidney disease, and there's a huge spectrum of disease that's associated with this. So here's this image. So essentially in the early 1980s, when the whole HIV epidemic occurred in 1981, I believe it was found, and then the wards that were created first here in San Francisco, 1984. So it was noted that a lot of Blacks were having HIV-associated nephropathy. So essentially, a huge amount of proteinuria that was found. And now we know that patients that have the APOL1 mutation had a 29-fold risk of developing HIV-associated nephropathy. So essentially, we can say, like, if the patient, to put it in simple terms, if someone has HIV-associated nephropathy, chances are they are Black and they have this higher risk variants. Now, since we have kind of passed all that with all the development that has happened in that world, it's more about the hypertension-associated ESRD or kidney failure that we're seeing this, but now we're learning again about the role of APOL1. It has been associated with all diseases, other things that are listed here, the transplant graft survival, which I'll touch upon later on. I was talking with some of the investigators in this area, talking also about there might be a role also in diabetic kidney disease, but this is kind of more disputed as well. So people that have the two risk alleles, so the ones that we typically see the disease, even though this is kind of changing now, tend to present with a significant amount of proteinuria. This tends to appear in the early 30s or so. In our building here at the VA, there was a young patient, African-American, that was with a creatinine of 2 and hypertoniuria, and he was found to have APOL1, the two risk variants of APOL1. So a little bit into the pathophysiology. So as I mentioned before, this is now found, the APOL1 mutation is now found in non-primates, and even in the primates, it's very limited, like I said, the chimpanzee. So this has been a big issue, a big limitation in learning about this disease. So in mice, again, this is not present, but when they have done kind of creating this mice, even those mice, so in the G1 and G2, they have the podocyte, the podocytopathy, but also those that with the G0, those that are not supposed to have podocytopathy also present with this, also have glomerulosclerosis. So it has been difficult with mice, but however, in 2019, 2020, Dr. Schuster, Kathleen Schuster from UPenn, did create a mice model that's being used now that kind of simulates more what we see in humans. But again, this is quite recent. In the zebrafish, they don't have, again, none of these animals have it. They don't have the APOL1. They do have a gene, a protein that's homologous to the APOL1, but for the zebrafish, it is important for the adequate glomerular function, which is different from humans. And in the Drosophila melanogaster, the fruit flies, essentially the studies that have done this mutation is completely lethal, so they all die. So this has been quite a limitation for us to understand more about the exact pathophysiology, what happens with this. But what has been proposed thus far, first of all, the A kind of extrapolating from what the trypanosoma does and the APOL1 protection, kind of opening cation channels in the lysosome. So that's kind of one of the what's been mentioned. Mitochondrial dysfunction and the plasma reticulum stress. Also, inflammasome leading to interleukin-1 beta activation, which I'll touch upon later on. Global suppression of protein synthesis as well. So some of the things that have been extrapolated from what happens with the trypanosoma, and trying to understand a little bit more about that, but this is an area that has been understudied thus far. So important to mention here that in 2016, there was a patient, a 54-year-old male in Ghana that presented with gastroenteritis. He has some nausea, vomiting, and he was found to have a trypanosoma infection. And the reason why, so he got the treatment, all that was done. He had HIV as well, but he was not on antiretroviral therapy. But the reason why an astute person kind of found this to be quite interesting is that they had not had a trypanosoma infection in over 10 years. Like, that's not something that is supposed to happen. And what they were able to find is that this patient, even though that he had the APOL1, he had a mutation here, the 264K, he had a mutation that kind of created a protective mutation from, which was later, I'll put it like this, a mutation during the APOL1. So he wasn't as protective for trypanosoma infection. So that's why he developed that. But later on, what we got to understand is that this mutation, even though it leaves the patient susceptible to trypanosoma, it does protect towards the development of kidney disease. So now we know that there is a variance. We have the G1, the G2, those that put a higher risk of kidney disease, but there is a variant, that N264K, that protects against kidney disease. However, kind of the patient is still susceptible to trypanosomiasis. So this study published in 2023, last year, using the Million Veteran Program, the MVP, obviously here with the VA. So they looked at a patient with African ancestry, and first they looked at those that had the mutation, the N264K, those that were positive, negative, and you can see clearly here in this panel, those that didn't have it had a higher risk of chronic kidney disease, and also of kidney failure or end-stage kidney disease. And then they used this in replication cohorts, and they were able to determine that he also had a protective factor. So those that had that mutation, that N264K, were protected against kidney disease. So we're learning more about different variants. There are more variants besides that G1, G2, and this one, but these are the ones that receive more attention, the more that we consider more clinically relevant. So we know that the G0 is present, the G0 variant is present around 50% of people. The G1, the combination, so two G0s, 50% G0 with G1 or G0 with G2, around 35% of participants, and around 13% of people have the two mutations, so the G1, 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 G2, or two G2 variants. Of those 13% of people that have the two high-risk variants, around 20% actually develop chronic kidney disease. So all that spectrum that I mentioned, the FSGS, progressing to kidney failure, only happens in around 20% of individuals. So like other kidney diseases, such as IgA nephropathy, other diseases that we have, so we have learned that there's kind of a second hit. Again, not everybody develops it. So people with one risk variant typically do not develop kidney disease. So the presence of the two risk variants, again, around 13%, but still do not develop the disease. So there must be something else. So we have learned, obviously, we go back to the 1980s, the HIV, as I mentioned before, the HIV-associated nephropathy, which is virtually non-existent in people without the APOL1 risk variants. We have since learned about the parvovirus B19, also with the recent pandemic, COVID-19, a lot of reports about people that, particularly African Americans, that develop kidney disease, different FSGS, collapsing glomerulopathy, and again, when they were tested, they had the APOL1 risk variants. So we know about also if patients that are receiving interferon or that have high interferon states develop this as well. And actually, interferon is what is used with many of these kind of mice models that's kind of injected to kind of create the injury. We are learning more about other genetic factors. So people that can have also the APOL3, that there's a physical interaction that can happen. We are learning more about environmental role, about pollution. So some studies published in 2020, 2021 by Nadkarni in Mount Sinai, kind of showing the role of that. We're also learning about the diet. Dr. Titi Ilori from Boston University is conducting a very neat study in Africa, and kind of people that have different diet, the Western diet, the kind of a vegetarian diet, and those, some of the preliminary results is not done yet, but in discussing with her, those with the kind of having more of a vegetarian diet had less development of this, but again, we need more data. But the point being, not everyone that has the two high-risk variants develop the disease, so we're learning more about other things that may happen as well. Recently, in April of this year, there was a conference by the KDIGO, by the Kidney Disease Improving Global Outcomes, so kind of the major organization in our field in nephrology. And they had this conference, and one of the topics that they discussed was that better understanding about the susceptibility. Who are the people that are going to develop this? Better monitoring. Who should be kind of focusing on more, targeting different therapies and things like that. So I mentioned that to say that in 2018, that currently the same investigator that I mentioned before noted in a cohort of around 700, 800 individuals that people that had three biomarkers, some of inflammation, some of injury, one of them was kidney injury molecule 1, tumoral necrosis factor receptor 1 and 2, had a higher risk of developing kidney disease. So people that had the two high risk variants and had the elevation of those three biomarkers were more likely to have this kind of renal endpoint of kidney failure. So we're learning about different biomarkers that may help us identify those patients that have a higher risk, again, for monitoring, for recruitment, for studies, things like that. Which kind of brings me trying to tie this up to a little to my research, working with Dr. X, who's here, Dr. Rifkin. Our primary goal has been understanding the role of the tubal health and progression of kidney disease, acute kidney injury. Our field, nephrology, is essentially it's very glomerulocentric. So many of the labs that you guys order that we have available here at the UA across the world, essentially the serum creatinine and albuminuria primarily can reflect what is happening in the glomerulus, right? So, but 90% of the kidneys is composed by the tubules, which we don't typically assess, or we don't really have a good ways to assess. So kind of our goal has been to understand what can the kidney tubules tell us about the overall health of the kidney, and if that is associated with outcomes. So I've published several studies kind of looking at different biomarkers, alpha-1, essentially studying different functions of the tubules, reabsorption, acid-base control, the protein reserve, different things, and trying to see if that is associated with progression of kidney disease. I did not mention this before, but it's important to note that the serum creatinine and albuminuria don't always correlate with the final pathway that happens in kidney disease, which is interstitial fibrosis and tubular atrophy. So this led to all these studies. So I've been able to identify, and some unpublished data here in some of my preliminary studies, that there are some biomarkers that that can tell us again which patients are at higher risk of progression of kidney disease. It was done in SPRINT, in the systolic blood pressure intervention trial, essentially a randomized trial that was to identify patients at high risk of cardiovascular events, to show if the standard blood pressure of 140 or an intensive arm of less than 120 was associated with cardiovascular outcomes. So using that cohort, there were around 94, 92 individuals that had the two high risk variants, around 200 that had one risk variant. I was able to identify and stratifying here between those that had 0 or 1 high risk variant versus those that had the 2 risk variants, and those that were more likely to progress or have an eGFR decline, even again, despite a very small cohort. So again, we're trying to understand, stratify those individuals on a higher risk of progressing in kidney disease. Now, moving on to treatment, kind of grouping this into the conventional treatment and the novel treatment. So I put this here. For every complex problem, there is an answer that is clear, simple, and wrong by Mencken. So essentially, okay, so if there is proteinuria, there's albuminuria, okay, just use RAS blockade. I guess most of the residents could have done that's a question. Everybody was saying, okay, just give him lisinopril, give him some RAS blockade, give him something like that, and that will take care of it, right? So that's kind of what we typically think about, or losartan, right? Okay, so ARB, ACE inhibitor, just give them that, okay, the conversation is over. Let's move on to the next thing, right? So it turns out, so this study, in ASCO, so the African American Study of Kidney Diseases and Hypertension. So in this study, investigators were able to look at, I don't have the picture here, but they were able to look at different people that received different antihypertensive treatments. So those that received RAS blockade, to be more specific, they received ACE inhibitors, and those that did not receive ACE inhibitors, and they stratified that by people that had 2 risk variants, the one that typically progressed, versus those that did not have the 2 risk variants, and essentially there was no difference. So giving the RAS blockade did not make a big difference in progression of kidney disease. Other therapies that we commonly use, those of you that have been in our clinics, that we use for membranous nephropathy and many other diseases, steroids, MMS, cyclosporin, also no significant difference. And they studied those that received they received cyclosporine on MMF and dexamethasone, essentially. And again, those with the two high-risk variants, the progression was still significant compared to those that had only 0 or 1 risk variant. So again, we still, those common therapies haven't been able to kind of pan out in this disease. So then the question, I presume someone would think about, what about the most common medication that probably you're already thinking about? Some of you may be thinking, I'm not going to give you a clue, but we'll get to that. So we go back to the panel D here, the role of inflammasome playing a role. So there have been studies using one that was published in JCI in 2021, using the mice that has been able to kind of replicate more what happens in humans. I'm kind of showing the pathway here, the apoptosis. And we know that a key component in the pathophysiology of APOL1 is the NLRP3 inflammasome activation. And we have come to learn that the SGLT2 inhibitors, so that's a group of medication that almost everybody gets now. The SGLT2 inhibition kind of modulates the NLRP3 inflammasome. So it's essentially the picture here, the beta hydroxybutyrate kind of inhibiting the NLRP3 inflammasome and potentially kind of stopping all the cascade and the apoptosis that happens in APOL1. So this hasn't been studied before. This is something that has been proposed. Trying to do this study has been difficult because essentially all the major trials that have looked at SGLT2 inhibitors that you are very well familiarized with and why we kind of love SGLT2 inhibitors, most of them only recruited the participants, only around 5% or less were Black. So this has been very difficult. And having access to this, again, these were done by huge pharmaceutical companies. So this has been quite a big limitation into studying this. But this is one of the areas, again, that I would like to look into further in a grant that I submitted not too long ago. Now, going into the novel therapies, kind of putting here the 3 steps, either working at the area of the gene and DNA, the transcript, the mRNA, or all the way at the protein. So starting with the protein, so this is where most of the research has been done. So there are some drugs looking at this. So the rationale is kind of blocking the toxic activity. We don't know the exact mechanism of APOL1 causing the injury in the kidney cells, but essentially using the APOL1 channel function as a surrogate for the toxicity due to APOL1. So a company called Vertex, they started the APOL1 program in 2015. By 2018, they had developed this drug, the VX-172. They kind of did first in vitro studies, and they were able to show some good preliminary data. They had this in 2020, and then last year in 2023, published in the New England Journal of Medicine, this drug the VX-147, now known as inaxaplin, kind of showing a significant decrease in proteinuria. This was a study, pretty small, only 16 participants. So that is remarkable. It got published in the New England Journal of Medicine with just 16 participants. And they were able to show that there was a significant decrease in the level of proteinuria. The median was around 50%. As always, there's one individual that didn't want to cooperate that actually increased. In regards to side effects, it was very well tolerated. But again, this was a very, very small study, only 16 participants. They are doing a bigger study right now, and they are recruiting more individuals for this study. And again, the side effect profile was it was very well tolerated, but again, very small study. Kind of going back, also using oligonucleotides to kind of modify the expression of RNA. So kind of using the same principle that's been done for other diseases as well, very early stages, and going a little bit back further using JAK-STAT inhibitors, kind of blocking the transcript, the inhibition of the transcription. And again, very early stages. They are recruiting. They're supposed to finish recruitment in 2026, in a couple of years. So again, we don't know yet about these drugs, but more is coming in the pipeline as well. Then kind of wrapping things up, one of the questions kind of then becomes, is there any value in testing? Some might say no. So as I mentioned before, and I've tried to be very clear about this, even the presence of the the high-risk genotypes, the G1 and the G2 in any combination, 13% of the population, particularly here in America, have it. Obviously, in Africa, it's higher than that, around 25, 30%. But here in America, around 13% have it. Only 20 to 25% of those individuals actually develop that. So, okay, so having the high-risk genotype does not equal developing kidney disease. And the other thing, some of the skeptical might say, okay, what is the point? There's no treatments available yet. Some might argue, yes, it can kind of lead some guidance in the diagnostic workup, all the things that we do, and then can be pointless if you can identify this, or kind of blaming everything on the patient with regards to the blood pressure, or giving them more ACE inhibition, and the common things that we do. So it can help in that area. And also, again, important for understanding for future treatments that could be developed. Again, we did not know about this disease. Learning about it since 2008, there were way more individuals. We were posting about this presence, but we're not doing much, and everything essentially has been delayed. And also in the role of the kidney in kidney transplant. So I don't know if anyone in the audience can recognize this individual here. Does anyone know who he is? Yeah, not so. Yeah. Oh, okay. I should have said those are below or under the age of 40. Let's put it that way. So they are older than me, Dr. Hepakowski at the back. So you should know that. But yeah, Alonso. I can name the whole screen. The ones in the back were kind of. I expected everyone to know the ones in the back: Scottie Pippen, Michael Jordan. Oh, okay. Pulmonary. Yeah. Okay. Good. You're better than me. All right. So that's Alonso Mourning. And one of the interesting things with him, so in the height of his career, he was in the Summer Olympics in 2000. He started noticing that he had a lot of swelling in his extremities, lower extremity edema, and he was feeling more fatigued. So he thought, okay, this is just related to kind of more rigorous training. Maybe I'm getting a little bit old. Again, he wasn't old, but maybe it's something that is happening with the training. He was then found to have FSGS. He was FSGS, and then kind of doing genetic testing, he was found to have APOL1. 3 years later, he received a transplant from his cousin, and he has been doing pretty well. So there's more about this that we don't know, and it can be quite remarkable understanding this. And again, this was at the height of his career, not like 50, 60-year-old. Again, someone that one would not expect this. So there are people that we simply don't know. We don't do enough testing. In regards to transplant, it has been shown already that people that received transplant of people of donors with the two high risk variants, that the transplant, the duration of the life of that graft is lower than those that have no risk factors, no risk variants. And there's a study occurring right now. They got renewed again, the APOLO study, funded by the NIH and two more institutes as well, around 260 centers. So they are doing testing in pairs, so both in the donors as well as the recipient, trying to understand more about this. So again, more and more learning. This can be problematic because again, it's a donor that has the APOL1 risk variants or higher risk of developing himself kidney disease, and this we will not know. Here at UCSD, talking with the transplant folks, Dr. Shah and all the group, they do test for APOL1 among African Americans who are interested in being donors. So we're learning more and more. And again, this study is not out yet. There have been smaller studies from the cohort, but again, they're still recruiting and trying to learn more about this as well. And this is what they have said for the Transplant Association. Those that have independent risk factor listed here, the age, smoking, obesity, they mentioned there that the testing might be considered. Again, there's no hard and fast rule, but this is kind of the areas that is getting attention. And we'll learn more in the upcoming years about this. At UCSD, you can order the testing. There's a private company that can do this. This is one of my patients, the other side of UCSD, that had one high risk variant, and they put here the gene APOL1. They mentioned the allele that this patient had, the G2, and they put here the risk allele, and they put here in the bottom the presence of two risk alleles confers a 7 to 30-fold increased risk of developing kidney disease compared to those with low risk APOL1 alleles. As I alluded to before, we are also learning a little bit more about those that have one risk allele in regards to developing hypertension. So, but again, we need more and more study in that area. So essentially, this has been kind of the beginning, going from 1977, 2010, 2011, understanding where it's located, and studies that I've mentioned since then. But going back about all this notion about this hypertensive nephrosclerosis, and kind of ending with what Dr. Friedman said. So the slide here, so he was already in 1993 mentioning this about the potential genetic role. Now going fast forwarding to 2015, kind of agree with the comment that he made here that I've highlighted, that we believe the term hypertensive nephrosclerosis should now be abandoned and replaced with either gene-based, for example, APOL1 associated with sclerosis or arterial nephrosclerosis, and precision medicine will be key to improving diagnostic accuracy in this field. So essentially where we are moving in nephrology, there's the KPMP, so the Kidney Precision Medicine Project, trying to identify and doing more biopsies, getting more genetic data. Here at the VA side, obviously the Million Veteran Program, the MVP, that was instrumental in identifying this variant, that N264K, that protects, that confers a protective role. So we are kind of moving more and more into the genetics and trying to tie that with biopsy results and moving forward. So kind of the key points, I would say, first of all, APOL1-mediated kidney disease is a key driver of kidney disease among people with African ancestry. And again, in one of the, you mentioned that, but in one of the studies, the one where they looked at MMF, cyclosporin, and dexamethasone, there were individuals that did not self-identify as Blacks that also had this high-risk variants, that they also progress faster. So even in people, so we know about the Hispanic heritage, Dominican Republic, other places. So we're learning more about this and all the mixture that has happened. So yes, it's important people with African ancestry and going beyond that, the testing. Be wary of hypertensive nephrosclerosis. This has been an area of a lot of debate in our field. I remember in 2018 or 2019, in our local conference that we have, Nephrology for the Consultant, that is led by Dr. Woodell, a very well-renowned pathologist, was talking about, like, we should not be talking about this hypertensive nephrosclerosis, but it's kind of pervasive, and you might see this in some notes and all that. This, okay, I don't want to be too controversial, but be wary of that. Understanding the role of genetic testing. I'm not saying that every single person has to be tested. We know that the most common cause of proteinuria is diabetes. But again, someone that doesn't have diabetes or has been very well controlled, recently diagnosed, going a little bit beyond the box, particularly, again, African ancestry. And we do need further studies for risk stratification and evaluating the roles of currently available therapies such as SGLT2 inhibitors, and the risk stratification, like I said, the role of biomarkers, understanding more about the disease. Again, not everyone develops it. And again, not everyone can get all the treatments. We know about the side effects and all the things as well. So some acknowledgement, I only put 2 people here. Obviously, this took more than 2, but as always, Dr. Rifkin, instrumental in everything since 2015, even in this presentation, and Dr. X as well. Okay, thank you.
All right. I'm supposed to moderate. I'm going to be instrumental in moderating the Q&A, and I'm going to repeat your questions. So please ask your questions.
Thank you. Really nice talk. I learned a lot. Question I have is, if you have 100 African American patients in your clinic, how many of those would have the APOL1 mutation? And those, if you have, again, 100 patients with APOL1 mutation, how many of those would actually develop FSGS by age 40?
Sure. So how many, if we took a sample of 100 African American patients in clinic, how many are going to have the high risk mutation? And of those, how many are going to have manifestations of disease?
So I would say, so I will preface it by saying it depends on which clinic. So in the primary care clinic versus a chronic kidney CKD clinic. So the primary care clinic, so the behavioral clinic, based on the studies, around 15%. So let's say of the 100, around 15 will have that. Those that will actually be having the 2 risk variant will be around 4 or 5 patients essentially that will be a risk of progressing and having FSGS and all that in the clinic. So people that get referred to us in the CKD clinic, obviously going to be more than that. But yeah, but based on the studies that we have so far, around 13-15% of those have the 2 high-risk variants, and then 20% of those that will develop. So for the regular clinic, yes.
Mark.
That was excellent. That was really, compelling story. Well, I had 2 questions, one therapeutic one and one diagnostic one. So first, GLP-1 agonists, one of the ways that they inhibit the inflammasome, probably known is through mitochondrial.
Yes.
Preventing mitochondrial DNA release is a thought in the growing area. Has anyone looked at mitochondrial targeted therapies?
Not yet. And
Okay, so
one.
I'm just
I'm
gonna
sorry,
repeat.
yes,
Has anyone looked, Mark Hepikowski asks, has anyone looked at mitochondrial targeted therapies since SGLT2's part of their mechanism is to change mitochondrial behavior?
Yeah,
Yeah.
unselfish.
Just curious.
So in the slide that I had about the pathway, so that one of the proposed mechanisms, the C, the one that was in the top right, regards to the mitochondria. So we do know that it's in the pathway, but it hasn't been studied yet. I'm not aware of any of those studies. But yes, so that's an interesting point.
And then the second quick diagnostic one is to identify, do we know if there is any variability in inheritance of the gene or development of the mutation by cell type? Or,
Is there mosaicism?
Is it, yes.
Yeah, so the second question is there some mosaicism about how this gene is inherited or expressed that it might explain maybe why the 80% don't get it and the 20% do?
Yes, that has been posed. So they are being studied by Dr. Freeman talking about that. So there is some mosaicism as well. Yes.
Thanks for a great talk. Of the patients who had the mutation who then go on to develop disease, that 2.3% or whatever that you mentioned, relative to those who have the mutation who don't develop the disease, has anyone looked at other potential drivers of maybe increased transcription of the APOL1? Like, do they have more frequent viral infections upregulating interferon signaling? Do they have more frequent AKIs, intermittent AKIs over their course of sort of their disease progression?
So
that's the question. Basically, what is the second hit that explains this low prevalence? Is it frequent exposure to interferon, or what is it that's happening?
So in regards to the interferon, I haven't seen any study about that. So, but AKI, we don't know much. I did look at AKI. That's one of my areas. So in Sprint, I did look at AKI to see if those that had the risk variant had a higher risk of AKI, but I did not find an association. I'm not aware of any study showing that association. There was one study that did show more AKI during the pandemic. People that had the high risk variant, they have more AKI. That was a study done at the VA. So that study did show that. But when I tried to do that, I tried to replicate that in Sprint. So there were some limitations. Primarily, I would say that the N was quite small. Only 90-something individuals, 92 individuals that had the APOL1 out of the cohort of 600 that I had with CKD. But again, all those are areas that are kind of trying to be understood more about why do some people progress versus others. And like I mentioned, the last 2 things that have been identified has been the diet, but we haven't published those results yet, and the environmental, the toxins and all that from Mount Sinai. But we're learning again more and more about that.
I'll ask a question unless there's something in the chat. Teach us how you go about explaining this to a patient and consenting them for genetic testing. What's that conversation like? And then how do you share the results back with your patient?
Yeah, that's okay. Instrumental in everything. So the way I tell the patients, again, like, we don't fully know. So if, like this patient that I mentioned before, 40, 50-year-old African-American female, no history of diabetes, had proteinuria, and had all the things. So I told her, like, I'm not fully sure why you have kidney disease. There's this genetic panel. So the result that I showed here doesn't only test for APOL1, tests for a whole host of diseases. So we have this panel available that can help us have some answers. And if it's this mutation that I think we have, I can refer you to UCLA, so where they're doing kind of recruiting patients with this drug, inaxaplin. So kind of telling the patient that there is a pathway if we do find this. Obviously, that some want to know, some don't want to know, like anything, but kind of explaining where we are and being honest with the patient that I cannot fully explain why you have this, your blood pressure has been very well controlled, and I don't fully believe in this hypertensive nephrosclerosis. So I just try to explain that to the patient. And then we do have a genetic counselors if they want to talk more and all that and how that may affect their kids. There are genetic counselors available so if they want to know more and all that. I do mention that, but I've tried to be transparent and tell them understanding this. So, you know, yes.
So great talk. Thank you for sharing all this really cool work. You mentioned that if a kidney donor has APOL1, then it has much worse.
Yeah. So they would not.
You understand that?
Yeah.
For the Zoom audience.
Sorry, I forgot. Yeah.
So they're coming down the fact that they're checking donors.
Yeah. Yeah, we are checking donors for that. So that will, and I don't want to speak for the transplant program. I don't know if they tell them like you cannot be a donor, or probably something like that. They might explain the risk, or they might kind of encourage the person not to be a donor. But we do know and all this about that donor may be at higher risk later on to develop the kidney disease. So I don't know if they, again, I don't want to speak for them. They tell, okay, you cannot be a donor, but they kind of encourage them and counsel them and all. Because those are things that you will not pick up in the typical things that we do. The 24-hour urine collection may look great. Everything else might look great, but you do know that this patient has this. But again, to tell them you cannot donate to your kid or something, I mean, I don't think that happens. So I don't know.
So
Yes.
this gene leads to podocyte necrosis?
Yeah.
And so are there any biomarkers of podocyte necrosis that are specific that can be picked up in those 20% who were developed, FSGS in future?
We
80% those who don't?
we have some markers that in the study that I mentioned is not completely specific to podocyte necrosis, but kind of injury inflammation that have been picked up. Yes. So there are some biomarkers that have been picked up recently. But most of the field before it was kind of more of the genetics and trying to find therapies and all that kind of where the whole area had been filled, all the interest was at. Now we're kind of trying to stratify and those are developing. But most of the attention was more into, okay, they get FSGS, let's find a drug to prevent this and all that. But not so much about identifying those that are higher risk. But that's kind of what has been happening more in the last years. And one of the areas with the biomarkers that I'm interested in looking at. But thus far, only 3 biomarkers that have been identified, have been studied. Yes.
Another question. Is an APOL1 mutant recipient of an APOL1 wild type kidney transplant, do they have similar outcomes as wild type to wild type? Or is there something about APOL1 in the host who's receiving the new kidney where they also have poorer outcomes?
Do you pay that?
If you get a kidney that doesn't have APOL1
Yes.
high risk,
But you do have
yourself
APOL1.
So if
that
you're
would be-
Alonzo Mourning and you get a kidney
Yeah.
that's
Yeah.
Scotty Pippins, then
Yeah.
Good job, Dr.
Yeah, that will be the Apollo study that will give us some answers there. I'm not aware of any data. If you have it and you get it from someone that doesn't, yeah, that's one of the things that they want to look at, getting more answers to that. Because we do know like how much you have circulating is not really indicative of that disease and all that. So the presence of it in other studies hasn't been associated with disease. So just having it circulating is not that significant. But yeah, that's one of the goals of the Apollo study, understanding that better.
Do they also have high risk for, hearing problems and hearing deficits?
No, I haven't seen any reports of hearing. I've seen reports of preeclampsia. I've seen reports with regards to opportunistic infections that they have a lower risk for that. They have higher risk for preeclampsia. That's an area that's getting attention, but I haven't seen any reports about hearing issues in people with APOL1. Kind of going extra beyond the kidneys, those are the areas that I've seen. Preeclampsia does. People with APOL1 do have a higher risk of preeclampsia, but nothing about hearing that I've seen so far.
Is there anything pathognomonic on the renal biopsy that you can say, oh, that's an APOL1?
Dr. Kennard asks if there's something pathognomonic about these kidney biopsies.
I haven't seen anything that would be pathognomonic about APOL1. Like if we were to look back and saying like without having any genetic studies, can you say like based on the morphologic findings, this is APOL1? I haven't seen any reports of that. Again, many studies haven't seen that.
David.
Sorry, so there's a lot of things I don't understand. I'm curious, like as far as like waking up a patient transplant or even a recipient, is there a genetic panel that's recently done, and is that part of the genetic panel? If it's done.
Is there a genetic panel for kidney transplant donors or recipients? Is there like a standard, we always have to do this panel, and is this part of it?
I'm not sure what panel they use. I will guess that they use the same panel that I use with my patients with the companies called Natera, Renasite. That's the panel that we typically use. So my guess would be that the transplant clinic orders the same panel there at the UCSD. Here at the VA side, all the transplants are done at Portland, and there's kind of works with hubs, and they do all the genetic testing. I've been in contact with them, so they do that genetic testing. But I don't know the name of the panel. I know that here I use that the VA is kind of more targeted, like you can test for APOL1. At UCSD, we do have the advantage. We can order the whole genetic risk factors of the known genes, and you can discuss that with them. But the company again is Natera, so they are the ones that do that. My guess is that they use the same panel, the same company.
Thank you, Dr.
No, thank you.
Change medicine brand round will be held here next week as well.
Thanks, everyone. Oh,
Thank you.
awesome. Oh, there was a question.
Great job.